My name's Vanessa Evans, and I did my research on somatic gene therapy. For an overview of gene therapy is defined as the aspect of therapeutic intervention which involves the introduction of specific genes in a patient cell with the help of vectors. We learned in lecture that in a cell it is the stretch of DNA or gene that directs synthesis of specific proteins for cell expression and functioning. The sequence of nucleotides determines what protein is to be produced, but when there is an aberration in the sequence, unusual proteins are synthesized, or there's no protein production at all. This can then lead to genetic disorders such as sickle cell anemia, hemophilia, and Alzheimer's disease. Gene therapy is able to correct this issue by inserting new genes into the patient's cells, repairing the defective genes, or completely removing the problematic genes. What is somatic gene therapy? So, somatic gene therapy uses the insertion of a normal gene into the DNA of somatic cells in order to compensate for the non-functioning defective gene. Somatic gene therapy delivers the normal gene into a somatic, also known as the body cell, of an individual. This allows for specific locations of the body to be targeted, but also limits the inheritance of the correct genome to future generations. Somatic gene therapy ensures that the alteration is temporary, meaning it's only for the patient. This is different from germline gene therapy, which permanently alters germ cells, which are passed down to offspring, meaning that the corrected gene is inherited to successive generations. In these two photos, you're able to see the cells that are used in somatic gene therapy, known as vector cells. In the photo on the left, you can see that the vector binds to the cell membrane, where it's then packaged in a vesicle. The vesicle breaks down, releasing the vector, and the vector injects the new gene into the nucleus, and the cell makes the protein using the new gene. How is somatic gene therapy done? For somatic gene therapy to be done, DNA needs to be inserted into a cell. This is when a carrier molecule called a vector, which we just talked about, must be used to deliver the therapeutic gene to the patient's target cells. This can be done through in vivo gene therapy or ex vivo gene therapy. In vivo gene therapy is in the body, while ex vivo gene therapy is outside of the body. In the left, you can see that in vivo gene therapy involves introduction of therapeutic DNA directly into the patient's body. First, copies of the therapeutic gene are inserted into viral DNA, liposomes, or in form of plasmid DNA. Then, the genetically altered DNA is inserted into the patient's body by cell-specific direct tissue injection. Lastly, inside the body, the inserted DNA is incorporated into the cells of the specific tissue it was injected into. So these cells now encode and produce the needed protein encoded by the inserted gene. On the right, you can see ex vivo gene therapy, which is done through genetic alterations of a patient's target cells happening outside of the body in something known as a culture. First, copies of the therapeutic gene are inserted into viral DNA, and cultured cells are infected with the genetically altered virus. Next, the target cells are removed from the patient. Then the cells that were grown in the culture have been genetically altered and are reintroduced into the body. Lastly, inside the body, the genetically altered cells produce the desired proteins encoded by the therapeutic DNA. What can somatic gene therapy treat? A journal article written at the University of Tennessee Health Science Center showed that somatic gene therapy is able to treat severe combined immune deficiencies, hemophilia, blindness caused by retinitis pigmentosa, and leukemia. In addition to these diseases, somatic gene therapy is also being used to alter tumor cells in the treatment of various types of cancer. Scientists are hoping that further research will lead to more efficient and cost-effective treatment of many different human diseases through gene therapy. In the photo on the right, you can see that the introduction of cells of individuals with genetic diseases can be used in bone marrow, lung cells, muscle cells, and liver cells. This is why gene therapy can be used to treat so many different types of diseases. Somatic gene therapy has many benefits, but there's also some risks that come along with it. One of these being an unwanted immune system reaction. Your body's immune system may see the newly introduced viruses as intruders and attack them. This could cause inflammation and, in severe cases, organ failure. Another risk is targeting the wrong cells. Because viruses can affect more than one type of cell, it's possible that the altered virus could affect additional cells, not just the target cells containing the mutated genes. This could lead to healthy cells being damaged, which eventually leads to cancer. An infection can also be caused by the virus. It's possible that once introduced into the body, the virus may recover its original ability to cause disease, which would completely defeat the purpose of the gene therapy. And there's also the possibility of causing a tumor. If the new genes get inserted in the wrong spot in your DNA, there's a chance that the insertion could lead to tumor formation. On the right, you can see some of the genotoxic risks that are associated with gene therapy. When it comes to somatic gene therapy, there's much fewer risks such as aging, cardiovascular disease, and cancer. When there's germline gene therapy, there's much more risks. That's why, as of right now, somatic gene therapy is the main form of gene therapy that's being used, and germline gene therapy has only been used on animals. As of right now, the gene therapy clinical trials underway in the U.S. are closely monitored by the FDA and the National Institutes of Health to ensure that patient safety issues are a top priority during research, so that these risks do not have to be a main concern when gene therapy is the only option of treatment. As for the ethics of somatic gene therapy, there are many topics that are still in question, such as how can good and bad uses of gene therapy be distinguished? Who decides which traits are normal and which constitute a disability or disorder? Will the high cost of gene therapy make it available only to the wealthy? Could the widespread use of gene therapy make society less accepting of people who are different? And should people be allowed to use gene therapy to enhance basic human traits, such as height, intelligence, or athletic ability? These are all questions waiting to be answered as somatic gene therapy becomes more developed, researched, and clinically trialed. As for the future of somatic gene therapy, a primary obstacle to broader application continues to be the immune response to delivery vectors and products of foreign transgenes. Right now, despite the great successes of many somatic gene therapies, there's still as many as 50% of patients who are currently excluded from the treatment due to pre-existing immunity to viral capsids. Luckily, recent advances and efforts currently in clinical trials have led to technological advances to circumvent this immune obstacle, such as engineering of modified capsids that evade pre-existing neutralizing antibodies. This will then allow for a much more broad pool of patients to be eligible for the treatment. Here you can see a timeline of somatic gene therapy. It started with oligonucleotide therapies, which had successful treatments in 2016 and 2019. Then there were CAR T therapies, which had successes in 2017. Then there was AAV transgene delivery, which had success in 2017 and 2019, and is continuing to being worked on today. Then there was gene editing, which discusses the in vivo and ex vivo therapies that I discussed earlier. And the goals for the future of somatic therapy are universal donor cell therapies, so genome annotation and functional genomics will be able to be used, personalized therapies, which would involve base and prime editing, and the ultimate goal is for common disease to be treated through somatic gene therapy. So why choose somatic gene therapy? Somatic gene therapy has been proven to have many benefits. First of all, the protein you're using is being authentically processed, and it is able to wipe out genetic disease before it can begin. This eliminates the suffering for future applications that many treatments usually have. The therapy is also efficient. It may only even need to be delivered once. It's also more affordable than many other treatments because one gene is able to give rise to a large number of protein molecules. Somatic gene therapy is also known as a treatment of a genetic disease that has no treatment. So by targeting the reproductive cells, such defects can be got rid of for good. Many people who suffer from genetic disorders such as Parkinson's, Alzheimer's, or Huntington's disease have gene therapy as their only hope. Because gene therapy has been able to help so many patients with all different types of conditions, there's great hope that it can evolve and potentially be the cure to many diseases that are currently known as uncurable.